Good morning, everyone. Thank you for joining me on this Science Week. My name is Dr. Neil Holden, and today I'm going to be talking about viruses. If there's one thing that I know over the last year is we've all become a bit more of an expert in viruses. I am a virologist. That almost sounds confessional, doesn't it? My name's Dr. Neil Holden, and I'm a virologist. So I did my degree in virology, and I've been studying viruses for my academic life, and my own particular research interests are in viruses that infect the lungs. But today I'm really going to talk about the basic biology of viruses. So what is a virus? So when I put this question out, I put some questions out on Twitter and other social media. The sort of questions I mostly get asked are things along the lines of, so what is a virus? What actually is a virus? How do viruses actually cause disease? Why don't antibiotics treat viral diseases? And do all viruses cause disease? Okay, and these are some of the questions I'm aiming to answer in today's session. Before we really delve into the sort of the biology of viruses, first of all, I just wanted to briefly remind you of some of the core biology concepts, because this becomes really important when we talk about how viruses actually work. So us as animals, and as indeed most life on Earth, we are made of cells, okay? So this is a typical animal cell, as you can see here. And there's major sort of architecture within these cells. So the cell machinery that exists within our cells are there to really do things like make proteins that make the structures that make up us, and generate what we call metabolic energy, so that we can actually carry out all those processes, so we can digest food, so we can do all the things that we need to do, grow and all these kind of things to actually be alive. So we also contain DNA. So DNA is our genetic material. And really, if you sort of fundamentally think about what DNA is the instruction booklet of how to make us, us. So DNA contains the instructions on how to make all of the different proteins within your body, okay? So DNA is copied into a molecule that's called RNA, and that RNA moves out to the cellular machinery in the cell, and that's what, and then the cell machinery binds to that RNA and makes the protein that RNA contains the instructions for. Okay. The other kind of life that we have on Earth, major piece of life, is bacteria. And we all know a little bit about bacteria. So bacteria are also types of cells. They're single cell molecules, organisms, and basically the biology of the bacteria is very different from our own cells. And that includes things like the membrane that surrounds them and the cell machinery that they use inside them. And that makes it very easy for us to actually design drugs which specifically target the bacteria. Okay, allows us to come up with antibiotics, and that the antibiotics either stop them from growing or it causes them to burst open. And so we kill the bacteria, and so they can no longer actually cause any infections. So what about viruses? Well, viruses aren't cells, okay? Viruses, as if you really define them, basically, they're what we call obligate intracellular parasites. So that means they are unable to replicate or do anything without infecting a host cell. So they're a parasite. They have to actually do anything, they have to enter a cell, okay? And that's really because they're unable on their own to generate metabolic energy. So they're not able to carry out any of those processes to actually undergo replication. They can't actually generate energy themselves. They also don't have any of the machinery to make their own proteins, okay? So viruses are fundamentally genetic material, and that can be either DNA, like our own cells, or they can actually contain RNA. So they can actually contain this other kind of genetic material, okay? And they're generally surrounded by a protein coat. And if you might have come across this term called the selfish gene, it's based on a book Richard Dawkins wrote a number of years ago. And fundamentally, if we were ready to think about what a virus is a selfish gene, okay? So basically, a virus exists to replicate. That's what they've evolved to do. In actual fact, if we really consider it, it's evolutionary disadvantageous. So it's not good for viruses evolutionary wise to cause disease, because if a virus is causing a disease, they're either damaging their host, and then damaging their host, they're not going to be able to use that host machinery as well to actually replicate themselves, or they're going to stimulate a host immune response, and that immune response is going to be targeted against that virus and is going to destroy that virus. But we're going to be talking a little bit more about the specific details of viruses and how they cause disease later on. Okay, so a little bit first about how viruses were actually discovered. I love history. I love delving into the history of science and things like that. My students know that I always throw a little bit of history into my lectures. So how were viruses discovered? Well, the word virus itself is from Latin, and it generally means a slimy fluid or poison. And prior to the sort of late 19th century, so the late 1800s, it was actually used as a non-specific term about any disease-causing organism, okay? But in 1884, there was the invention of a very, very particular kind of filter. Now, this filter was made out of porcelain, and the holes in this filter were so small that it stopped any kind of life from going through. So bacteria couldn't pass through it, any kind of sort of fungus couldn't pass through it, any kind of organism got trapped in the filter, and everything else passed through the filter. Now, around the same time, a scientist known as Adolf Meyer was investigating a disease of plants, okay? And this disease was of a particular plant that was very important economically at the time, which was the tobacco plant. Now, this disease is called a mosaic disease. So you can see here on this diagram, on the left-hand side, this is a tobacco leaf that is being affected by this disease. And on the right-hand side, we have some unaffected leaves. So Adolf Meyer was investigating this disease. Now, a couple of other scientists at the time brought these two discoveries together. So Dmitry Ivanovsky and Martinus Beijerinck, sorry, I always stumble over some of these names. Actually, both of these scientists did a very similar experiment. So they took some leaves that had been afflicted by this disease, and they mashed them up, and they passed this sort of mash up of leaves through this filter, okay, this filter on the left here. And basically they took everything that came out of that filter. So remember, bacteria and all those kind of things would be trapped in the filter, everything else would pass through. So they took this liquid that came through the filter and put it on some fresh tobacco leaves and found that it still got the disease. So whatever was causing the disease was able to pass through this filter, okay? And they were called filterable agents. Now, Dmitry Ivanovsky thought it was a toxin, so some sort of chemical was actually causing this disease. Martinus Beijerinck actually thought it was what he termed as a living fluid. So he thought that it was actually the liquid was living, it was causing disease, and it was actually this that was causing the damage to the tobacco crop. So both of them were a bit wrong, but this was the first step in actually discovering the virus. They also did some similar experiments around the same time in animals too, and particularly in a very, very sort of prevalent disease in some farm animals called foot and mouth disease. Okay, so foot and mouth disease particularly affects cattle, and it can go through herds very, very rapidly and ultimately cause major damage to farming communities. So again, a couple of scientists, Paul Frosch and Frederick Loeffler, did very similar experiments. They took samples from cattle that were suffering from this foot and mouth disease and passed it through again one of these filters. They took the liquid that came through the filter and showed that it was able to still cause the disease. Okay. They did something very interesting though. They treated the fluid with some heat. So they heated up the fluid and then they tried and injected that into some cattle and actually showed that this heated up fluid, not only did it not cause the disease anymore, but it actually protected the animals they'd injected. It protected those animals from foot and mouth disease. So they had actually created a vaccine. And this vaccine is the first use of an inactivated virus as what we call a prophylactic vaccine, as a protective vaccine that was actually used to treat and to help prevent disease in these animals. And over the next 20 or so years, many viruses, particularly ones which infect humans, were shown to also be these filterable agents. We still didn't know exactly what they were though, okay? So one of the diseases was yellow fever. Another one is polio. So this is actually a hieroglyph found in Egypt, and it shows this withered leg, which is very, very typical of polio infection. So polio is a very, very ancient disease. We've known about it a long time, but it wasn't until the 20th century that we actually found that it was potentially caused by these filterable agents. There's a disease in cattle called rinderpest, which again wiped out herds of cattle, was also found to be caused by these filterable agents. Smallpox and rabies. So these were all diseases that majorly affect either humans or animals that were found to also be caused by these agents that were able to pass through these very fine filters. So in the 1930s, we first got our glimpse of what these agents were. And that was thanks to a couple of scientists called Ernst Lusker and Max Knoll, who invented the electron microscope. So suddenly we're able to see things that were much, much smaller than we're normally able to see over a light microscope. So a normal microscope just isn't powerful enough to see viruses. So we need an extra powerful electron microscope to actually visualize these viruses. And one of the first things that's actually done, it was actually done by Ernst Lusker's brother, who was also a scientist, is they started looking at these filterable agents and kind of identifying them and sorting them and classifying them depending on their shapes and sizes. And we know that viruses come in all sorts of shapes and sizes. Okay, so we know they can come in very sort of filamentous, worm-like appearances. They can appear very structured in an icosahedral kind of shape. So almost like this very sort of structured shape made out of individual triangles, as you can see here. They can be called what we term pleomorphic. So pleomorphic means that they're kind of flexible. They're generally round, but they can be a bit stretchy, be a bit oval, be a bit sort of stretched out, and all these kind of things as well. Or they can have very, very complex structures, as we see here with bacteriophage. And bacteriophages are viruses that we'll talk about in a little moment. And these are have very, very unique structures associated with them. So they have these heads and tails, and it almost looks at what I always think about when I see these viruses is they look a bit like lunar sort of landers. So that, the spacecraft that landed on the moon kind of thing. But we know they come in all sorts of shapes and sizes. So you can see in this diagram down here, this is actually to scale, as it were. So we have some larger viruses, as you can see up here, some very sort of worm-like viruses, and some very, very small viruses compared to the others. Okay, so here's some actual pictures of viruses. This is probably one of the most famous pictures of a virus. This is the Ebola virus. And it's a very sort of worm-like appearance. It's often referred to as a shepherd's crook. So imagine a shepherd and the stick they use walking around. these viruses are very sort of worm-like, but they can be very structured, as you can see here and here as well, sort of very round like. So how big is a virus compared to us? We know they're small, but just how small are they? So a virus typically is around 100 nanometers or so in terms of its diameter, okay? So it's very difficult to sort of picture what 100 nanometers is. So, you know, if we take a millimeter and we divide that into 1,000, that's 1 micrometer. And if we take that micrometer and we divide that into 1,000 pieces, then that would be 1 nanometer, okay? But if we took a virus and we expanded it up so that it was as big as a drawing pin, for instance, as you can see here. So a drawing pin is typically around 5 millimeters or so across. So if we made a virus, the diameter of a virus 5 millimeters across, well, bacteria, so bacteria generally is around 6 micrometers in terms of its size. Bacteria would be about the size of a dinner plate. So if a virus was as big as a drawing pin, a bacteria would be about the size of a dinner plate, so about 30 centimeters in diameter. So what about a human? How big would a human be? Well, of course, I have to pick a human to use on this. I used to use Danny DeVito, but my daughter strongly suggested I updated my reference, and so we use the Ariana Grande. Okay, so Ariana Grande is our reference human here. Ariana Grande is around 1.52 meters, so just over 5 foot tall. And so if we size up a virus to the size of a drawing pin, bacteria size of a dinner plate, Ariana Grande is 76 kilometers high. Put that in perspective, if she laid down in the UK and used Hull as her pillow, she would be resting her feet in Lincoln. Okay, so viruses are tiny. Okay, compared to us, they are absolutely minuscule molecules. But how many are there? So how many viruses are there? We know they're small and they can fill up a lot of room, but there's actually more viruses on Earth than there are stars in the known universe. Okay, so there's lots of viruses. That's the total number of viruses, not the total number of kind of viruses, just the total number of viruses. The estimate is around 10 quadrillion viruses. That's a 10 with 30 zeros after it. If we took all the viruses on Earth and stacked them end to end, they would stretch around 100 million light years. So that's enormous distances. If we took a litre of water from any sort of coastal sea, so if we took a litre of seawater from anywhere near the coast, that litre of water would contain more viruses than the number of humans on Earth. So there are a lot of viruses. In terms of ones that we're particularly interested in terms of humans and disease of animals and these kind of things, we know there's approximately 320,000 types of virus that affect mammals. So in mammals, in sort of that whole kingdom of mammals, there's probably around 320,000. Now, we don't know the exact number because we haven't detected them all. And there's, we know that, some species, particularly bats, are more susceptible to having viruses within them. And some species, we still don't know how many viruses there are. We haven't necessarily discovered all the animals on Earth, so we won't have discovered, until we've discovered all life on Earth, we won't have discovered all viruses. One thing we can say is it's almost inconceivable that there is a form of life on Earth that isn't affected in some way by a virus. So in humans, we know there's around 219, 220 different viral species that affect humans, but we're discovering around 2 to 3 new viruses every year in humans. And as we know, in this last year, we've had a new virus move over into humans, which was the SARS-CoV-2, which is the causative, which actually is the name of the virus that causes COVID-19. Okay. So we know we've got viruses. We know viruses are causing disease. We know they're very, very small. We know that they're what we call obligate intracellular parrots. They have to infect a cell to actually replicate. So how do they do that? How do they actually replicate? So for a virus to enter a cell, so here we have here in our little grey round sort of circle here we have a virus, and here we have our animal cell. Now, on the outside of the cell, on all of our cells, we have what we call receptors. So these receptors are proteins which are there to bind to signaling molecules and respond to things in the environment around the cells. Well, viruses have evolved to actually take advantage of these receptors. So the first thing that a virus needs to do to infect a cell is it needs to attach to that cell. So a virus will enter an organism. So let's take a human. A virus will enter a human, and it will find the cell for which it can infect, and it will bind to a receptor on the outside of that cell. Now, one of the natural responses of things binding to receptors is that cells will engulf those receptors and take those receptors inside themselves. So we see that happening is that the membrane that surrounds our cells will surround that virus, and it will engulf it and bring it into the inside of the cell in a little sort of bubble, okay? And these are called vesicles, okay? So the virus then is able to escape that vesicle. It's able in some way, and to be honest, having studied virology for years and years and years, there are so many different mechanisms by which the viruses do this. So this is a very generic overview of how viruses replicate. Viruses are extremely diverse, okay? So viruses can actually replicate in many, many different ways, and they undergo these steps in many, many different variations as well. But generally speaking, a virus will need to escape this vesicle, okay? And normally, in some way, it will escape out of that vesicle. So once inside the cell, the virus will take off its jacket. You know, it's inside, it's coming from the cold or whatever, and the first thing it's going to do is going to take off that protein jacket. It's going to shed its protein coat that surrounds that virus, that gives that virus its structure. Now, inside that protein coat, we find the genetic material of the virus. Now, again, this genetic material can be DNA or it can be RNA, okay? So, as well as this genetic material, a number of the viruses will also bring into it proteins. And these proteins are things like enzymes and other molecules that the virus can use to help it take over the cell. So the virus genetic material will either move into the nucleus, where our own DNA is found, or it might actually stay in the cytoplasm. So very, very generally speaking, DNA viruses move into the nucleus. RNA viruses kind of stay in the outside parts of the cell, what we call the cytoplasm of the cell. At the same time, the virus proteins are binding to and taking over the cellular machinery. So this is the machinery, remember, that generates metabolic energy, that actually manufactures the proteins and things like that. So a number of viruses will actually subtly alter our own machinery in our cells, so it stops making proteins for our cells and starts only making the proteins for the virus, okay? And so basically, it starts replicating, and it starts producing lots more of the genetic material of the virus. And at the same time, it's changed that cellular machinery, so it's making more viral proteins. And that includes more of the viral protein coats, what we call the capsids of the virus. So the virus is replicating, it's using the machinery inside of our cells to help make more of the virus. And that's really why we term a virus as a parasite, because it's using us, it's taking over our own machinery to actually replicate and make more viruses. Ultimately, the virus will then escape from the cell and go on to infect other cells. It can do this either through budding out of the cell. So sometimes a virus will just leave the cell. It will actually pinch off a piece of the membrane that surrounds the cell, what we call the cytoplasmic membrane. It will use that and it will exit the cell, taking a bit of that membrane with it, and that membrane is kind of surrounding the virus. Or the virus will actually overwhelm the cell, either through taking over the machinery or just through the sheer numbers of virus within that cell, and it will cause that cell to just burst open. And when the cell bursts open, it releases all those newly made viruses into the environment, and that virus can then go on and actually infect other cells close by. So how do viruses actually infect us? How do they get inside us? Well, viruses are all around us all the time. So basically, how do viruses actually do that? Well, we've got a number of openings into our bodies, natural mucosal openings. So we have things like our eyes, our mouth, and sort of our guts, so our stomachs and intestines, our nose and our lungs, our sort of genital tracts and our bums and all these kind of areas. These are all ways in which viruses can actually enter the human body. So they've evolved ways to actually pass through these mucosal membranes that we find in our eyes, in our mouth, and our nose and airways. We know, for instance, when someone has a cold and they sneeze, and another person could breathe in that particles from the sneeze that contains the virus, and it will enter either through our nose or through our mouth into our lungs and infect those cells in our lungs as well. We can also have cuts and abrasions. So our skin is generally very good at keeping out viruses, but obviously if we damage our cuts, so if we have a cut in our skin, that also gives an entry point into our body to pathogens, to viruses and things like that. But also if we have things like injections. So if someone injects themselves with a contaminated needle or something along those lines, that can also directly introduce a virus into our body. So the virus is entered our body and it's replicating in our cells. So how does it actually cause disease? Why do we get sick when a virus does that? There's a number of different ways viruses can actually cause disease. So firstly, the virus can actually damage the body by directly killing those cells. So this is actually some pictures that I took in my laboratory. So this is some cells that we can see down the microscope. These are lung cells. So these are what we call the epithelial cells from the lung. And on the left-hand side, you can see some nice, healthy epithelial cells from the lung. Now, what I did to these cells is I infected them with my favorite virus. I know you've probably all got your own favorite viruses, but my favorite virus that I like working with is called respiratory syncytial virus. And when I infected these cells with respiratory syncytial virus, you can see here on the right-hand side just how much damage that virus infection has actually caused. It's killed off huge numbers of those cells, and it's just really destroyed those cells. So viruses can cause damage, and by killing off those cells, by causing enough damage in particular tissue, they can actually then cause that tissue to be damaged. And ultimately, for that, if it's doing it if it's destroying cells in the liver, well, ultimately that liver's not going to be able to work as well, and therefore we might start seeing symptoms of a damaged liver. The second way is viruses can actually cause genetic changes. So some viruses, when they inject their genetic material into our cells, that genetic material can actually insert itself into our DNA, and it can basically sit inside the DNA, and it can also do this in bacteria and other species of life. And basically, it can cause a genetic change. So if it inserts itself into something that's really important in our DNA, a gene that's particularly vital for some functioning of our body, if it disrupts that gene can no longer be made, and it can ultimately cause disease. The third way is it's actually not the virus that's causing the disease. The third way is it's actually the immune system and the immune system response to the viral infection that causes disease. And we classically see this with viruses like, viruses that cause the flu, so the influenza viruses that cause common colds. We see symptoms like having a runny nose, having a fever, having body aches, having a sore throat. All of these symptoms are actually being caused by the immune response to the viral infection. It's not the virus itself causing these symptoms, it's actually our own body. So for instance, a runny nose, having lots more mucus. Well, the body's making more mucus so they can try and trap those virus particles. Mucus is very great, it's good sticky material. And if viruses come in contact with that mucus, they'll get stuck to it like a fly to fly paper. And basically they can be removed from the body quite easily in that mucus. A fever is our body increasing the temperature of our body so the virus is unable to replicate as efficiently as it does. So viruses have evolved to work best at our normal body temperature. If we raise our temperature, those viruses are not going to be able to work as well at that increased temperature. And it's actually this third mechanism that causes the vast majority of symptoms in viral disease. So it's actually the immune response that causes a lot of those symptoms. So why don't antibiotics work on viruses? Well, antibiotics, as I said before when I talked about bacteria, they specifically target the bacterial mechanisms, the bacteria machinery. And to be safe, they have no effect on the animal cell machinery. So they don't have any effect on our cells. An ideal antibiotic would have no effect on our own cells. Well, we know that viruses take advantage of our own machinery. So because the antibiotics only target the bacteria machinery and have no effect on our cellular machinery, they're going to have no effect on the virus itself. So the virus, by being that parasite, by taking over our own cell machinery, means it's a lot harder to target viruses with drugs. So why don't we have many antivirus drugs? Why can't we have the equivalent of an antibiotic for viruses, an antiviral drug? Well, the problem is, most of the time, by the time you actually start really exhibiting symptoms of a virus infection, that virus infection has been ongoing for some while. And it's actually, the symptoms don't develop until after the virus has really established an infection inside the body. So by then, it's pretty much almost too late to start taking a drug because the virus has already, it's replicated. And in many cases, it's actually our own immune response that's causing those symptoms. And that immune response is actually taking care of that viral infection. The other major issues is that viruses, because they use our own cell machinery, if we were to target that, it would actually be toxic to us. So because the viruses take over our cell machinery, if we were to inhibit that machinery, it would actually inhibit our own processes and cause probably just as much disease as the virus does itself. Viruses are also rapidly mutagenic, so they mutate all the time. They're a lot more easy to mutate than even bacteria. And so they quickly mutate and gain resistance to drugs. This was particularly seen in terms of things like HIV. Treatments to HIV, when they were first used, were very ineffective because HIV is so mutagenic. It's very, very expensive to actually develop viruses because viruses are so difficult to study in the laboratory. We also have this problem with toxicity and viruses with a high mutation rate. And actually what we found is that with viruses like HIV that are so mutate so much, the actual best way to attack these viruses is to use combinations of drugs. So if they gain resistance to one drug, you've got other drugs there to help deal with that virus. And that's actually become one of the most effective antiviral treatments out there in terms of drugs that we use. So I mentioned there that it's very expensive to study viruses. Well, that's because to actually grow a virus in the laboratory, we need to actually infect a cell. And you might think, well, why is that a problem? Well, for many, many years, we weren't able to grow cells in the laboratory. Particularly, we weren't able to grow human cells in the laboratory. Now, viruses are very, very specialized. So a virus that infects humans will grow in human cells. It will find it very difficult to grow in cells from another animal. You won't be able to grow a virus in bacteria that's adapted for human cells. So it wasn't until the 1950s that we actually were able to grow human cells rapidly within the laboratory. And this was in an unfortunate way, thanks to a lady by the name of Henrietta Lacks. So Henrietta Lacks, unfortunately, in around her 30s, developed cancer, and she developed cancer of the cervix. Okay, so she had cervical cancer, and basically and she unfortunately died from that cancer. But some scientists took some cells, and they actually took these cells without her consent or her family's knowledge. And this has been quite a controversy since then. But some scientists took those cells, those cancerous cells, from Henrietta Lacks. There was a scientist called George Gay, his wife Margaret, and their assistant Minnie, and they took those cells and they were able to grow them in the laboratory. Okay, and these cells are now called HeLa cells after Henrietta Lacks, and there's actually tons, literally tons of these cells grown in laboratories around the world these days. So Henrietta Lacks has left us a massive legacy in terms of able to study viruses. This was the first human cell line that was established so we could actually study viruses in detail. And now we have many, many different kinds of cells that we can grow in the laboratory, and we grow it in something called tissue culture, and we can grow cells in these plastic flasks. And as you saw in my earlier slides, some cells of those lung epithelial cells is what I do in my laboratory. And we can actually grow viruses in these cells, but it's all, if we were to trace it back, it really is thanks to people like Henrietta Lacks. And basically, these cells are immortal, and they're immortal because they're cancerous cells. So they just keep on dividing and dividing and dividing forever and ever and ever. And so this has really helped us learn a lot more about viruses. Finally, I just wanted to finish on a little part, just considering. So we talked about viruses. We've talked about how they can cause disease, about how they're hard to treat, about how they're hard to study. But can viruses be useful? And I want to put a couple of different scenarios to you guys. So can a virus ever actually be useful for us? Well, the first set of viruses that could potentially be useful, I mentioned them briefly earlier, are these viruses that infect bacteria. So there's a number of viruses that can infect bacteria, and they're called bacteriophages. Okay, and they were discovered in the early 20th century. So Frederick Twort, he was studying a particular bacteria called Staphylococcus, and suddenly he found that all of these Staphylococcus bacteria that he was studying in his laboratory were dying, and he didn't know why. And he took this culture, this liquid full of these dead Staphylococcus, and he did the same experiment as we saw earlier. He passed them through a filter and showed that it was a virus that could pass through a filter and cause more damage to other bacteria. And around the same time, Felix here discovered a virus that killed another kind of bacteria called Shigella. And he named it a bacteriophage. And bacteriophage literally means bacteria eater. Okay. And what he discovered is if he took this virus, which infected Shigella, and he injected it into rabbits, it protected those rabbits against infections by this Shigella bacteria. This gave first the idea that we might be able to use viruses as a replacement for antibiotics. So around the same time, antibiotics were also being discovered and being developed. And in some countries, they actually went down this bacteriophage therapy route. So instead of using antibiotics to treat bacterial infections, a number of places were looking into using these bacteriophage viruses. And now we're moving more and more into the sort of era of more and more bacteria being resistant to antibiotics. This is something that is being more and more strongly considered to be moving towards potentially using bacteriophages as a therapy to treat bacterial infections. Okay, what about other things? So is there other ways that viruses can be useful? Could they be used to treat cancer? Well, one thing we know is that some viruses actually contribute towards cancer. They actually cause cancer. So viruses contribute to around 15% of all human cancers worldwide. So they can cause things like liver cancer. So some of the hepatitis viruses can result in liver cancer. They can cause cervical cancer. So things that cause, some of the papilloma viruses, the HPVs, can result in cervical cancers. They're also a cause of some leukemias as well. But if we can actually genetically engineer viruses to just target cancer cells, so if we can actually make a virus that will only infect cancer cells, and as I said before when I talked about the replication, one of the ways that viruses can exit cells is by bursting them open and killing those cells. So if we can design a virus that just targets those cancerous cells and destroys the cancer cells and leaves our own normal cells alone, potentially we could have a virus that destroys just the cancerous cells and ultimately help with treating that cancer. Viruses could also be used to treat genetic diseases. So we have a huge number of genetic diseases. So people who have a particular gene that causes some sort of disease. And as you can see here, I don't expect anyone to sort of read these diseases. This just illustrates just how many different human genetic diseases there are. Okay. But again, if we can genetically engineer a virus, if we can manufacture a virus that we can insert the actual healthy gene into that virus and actually infect people with the genetic disease with this virus that contains the healthy gene, there is the potential that, again, remember that a number of these viruses, they insert their genetic material into our own DNA. So if we can get a virus to insert the healthy gene into our own DNA, then that might result in helping cure genetic diseases. And really, if we are to consider viruses as a whole, viruses have really helped shape us. Viruses have been an absolutely essential part of evolution of life on Earth. So viruses are a driving force of evolution. And that's partly by potentially causing disease and driving, survival of the fittest, as I say here on this slide, very much so. But also, viruses have become an integral part of ourselves. We are all part virus. So one thing I mentioned earlier is that, again, I've mentioned it a couple of times, is that viruses are able to integrate their genetic material into our DNA. And this has happened millions of times throughout our evolution. And when we actually first sequenced our own DNA, we found many actual pieces of evidence which showed ancient infections by viruses that have integrated themselves into our genetic material and have actually, lie dormant within our own DNA. They've become a part of us. So the virus has inserted its genetic material into our own DNA, and that it's just become a part of us. And in fact, there's been some really fascinating studies that have shown that a particular viral gene that's actually part of what we call the envelope gene of viruses, very similar to what we very sort of related to HIV, is absolutely essential in the formation of the placenta in sort of mammalian birth. So the placenta, the part which actually interacts, sort of the interface between the uterus of the pregnant woman and the fetus, that placenta layer actually potentially contains a virus gene. So viruses have really helped shape the evolution of life on Earth. Okay, so I would like now to invite you all to interact a little bit. So obviously, if you do have any questions, feel free to pass them. But I would like to invite you to play a game that I like to call "What's That Pathogen?" Okay. If possible, could you, if you have another device available, or you can post if you're unable to go to this particular website. So it's www.polleverywhere. One of my colleagues has put it into the chat there, and you should be able to vote on what's that pathogen. Okay, so I'm going to read out a number of diseases and illnesses, and you have to decide, is that a virus, a bacteria? Is it caused by both viruses and bacteria, or is it caused by neither? Okay, let's see how this works. Okay, so I've got, so if you go onto this website, you should be able to vote on what you think causes this disease. Okay, so the first illness is the common cold. So is the common cold caused by a virus, a bacteria? Is it caused by both, or is it caused by neither? See if I get any responses. Okay, we have a couple of people potentially saying on the poll everywhere. And you're absolutely correct. That's a virus is what causes the common cold. The virus actually most commonly causing the common cold is called the rhinovirus. So rhinovirus, obviously being named rhino after the Latin for nose, causes the common cold. And you might think, well, why do we get so many common colds? You know, why do we always keep on getting colds? Why doesn't our immune system protect us against the common cold? Well, unfortunately, there is actually near enough 200 different variants of the human rhinovirus. So there's around 200 different forms of that rhinovirus. And on top of that, other viruses, such as one of my favorite viruses, respiratory syncytial virus, also causes symptoms very similar to rhinovirus. There's a huge number of viruses out there that cause the common cold. Okay, so this is why we keep on catching colds. Okay, what about malaria? Does anyone out there know whether malaria is caused by a virus, a bacteria, both, or neither? Okay, we've got a couple of different responses coming in. So one person says virus, and I can't see, by the way, if you do vote on this poll everywhere forum, I can't see who's voting for what, just to reassure you. I can just see the total number of votes per option. So one or two people have said virus, and a couple of people have said neither. It's actually the ones who voted for neither are correct. So malaria is actually caused by a protozoa, an even slightly more complicated form of life. and it's actually neither a virus or a bacteria. Okay, so we'll just do maybe one or two more of these. So mad cow's disease, also known as bovine spongiform encephalopathy. It's one I always stumble over that one a little bit. So it's bovine spongiform encephalopathy, or mad cow's disease. Is that caused by a virus, a bacteria, both, or neither? I'm just looking at my other screen here to see those responses coming in. And you're all absolutely correct, those of you who voted. It's actually neither a virus or a bacteria. Diseases such as BSE, or mad cow's disease, in humans, we have Creutzfeldt-Jakob disease, CJD, is actually caused by an even simpler disease-causing agent. This is caused by what we call a prion. And a prion is just simply a protein. It doesn't even contain any genetic material. And it's a protein that has folded up incorrectly. And by folding up incorrectly, it actually then causes other proteins surrounding it, other prion proteins that are around it, to also fold up incorrectly. And they all start clumping together and causing damage. And we find prion proteins particularly prevalent in our central nervous system, in our brain. And they start clumping together and they cause holes to form within the brain. And that's why it's called a spongiform encephalopathy, is because it basically makes that brain tissue look very sponge-like, full of holes, as it were. Okay, finally, let's have a very sort of generic one. What about throat infections? So are throat infections caused by viruses, by bacteria, by both, or by neither? Let's have a look. And we have a bit of a diverse one here. So we have a number of you saying viruses, and some saying bacteria, some saying both, and some saying neither. And again, you are absolutely, majority of you are correct, and it is actually mostly both. Okay, so both viruses and bacteria can cause throat infections. Actually, 70% of all throat infections are caused by viruses. So we have viruses cause a vast majority of throat infections, which is why we need to be really careful about when we have things like throat infections, whether we need things like antibiotics. So the vast majority of throat infections are caused by viruses, along very similar lines to what we've seen already in terms of rhinovirus, respiratory syncytial virus, influenza. All these kind of viruses cause throat infections, but also we have things like strep throat, so streptococcus bacteria and Haemophilus bacteria and things like that. Okay, I had a big long list of other illnesses, but I am running a little bit low on time. So I would just like to thank you all very much for listening to my talk. If you've listened to it live, thank you very much. If you're watching this later on YouTube and you want to contact me with any questions you might have, you can find my email address on the University of Lincoln website. It's just nholden, that's NHOLDEN, @lincoln.ac.uk. So if you have any questions about viruses in general, anything that you want to know about anymore, if you've got any questions now, you can feel free to put them in the comments or contact me, directly, and I can email you any sort of my responses and things like that in terms of those. But thank you very, very much for listening, and have a great day. And I hope you enjoy some more of our Science Week talks later on.